We're really excited today to also welcome two additional new members that are going to present their research to us, Sha Yang and Ziva Cooper. I also would like to draw your attention to next week's Magoon lecture, which is given by Michael Sofroniev. And usually after the Magoon lecture, we have a little party that, you know, with food and wine and so on. So I invite you this year to do that on your own, to just come to the lecture with your own glass of wine. And in contrast to the lecture hall, you can actually bring in your coffee or your wine or your water or tea directly to the lecture and be happy during the lecture itself. So without further ado, I would like to introduce to you Sha Yang. Sha is coming from Shandong University, did her undergraduate at Shandong in China, and then got a PhD at Georgia State before she came and for a postdoc at UCLA. Then you say, oh, she came for a postdoc at UCLA and stayed ever after, but no, she actually went out to industry and worked for various companies in industry before she came back. I think it was 2011 to join the Department of Molecular and Integrative, yes, I got that right, Molecular and Integrative Biology at UCLA in the college, and rose through the ranks from assistant professor to associate professor. And she will talk to us about the multi-tissue, multi-omics systems biology of complex diseases. Sha?
Hi, thanks, Felix, for that kind introduction. I'm really excited to be here today to join the BRI, and I look forward to productive interactions in the future with everyone here. So I will mainly introduce the overall research scope for my lab. So as Felix introduced, that I have been in different places and studying different things over the years. So our lab actually study a broad range of complex diseases, ranging from cardiometabolic diseases to various brain disorders. So as system biologist, I look at bigger pictures. So the main idea here is that there could be crosstalks between different diseases. For example, we know the various cardiometabolic diseases are connected, and there are significant metabolic and immune components shared across the different diseases. So the idea is to look at the connections, not only understand individual diseases, but look for connections across diseases. So we take a hybrid approach from big data integration to experimental testing. So the idea of using the system biology is to truly understand the various dimensions of these complex diseases. We know for each of these diseases, both genetic and environmental factors influence the disease susceptibility, and these various risks can affect various tissues. And within each tissue, there are many different molecular layers, from the DNA to the genome, to epigenome, transcriptome, proteome, metabolome, etc. And also these different molecular layers don't just act alone, they interact in this multi-tissue networks. So not only there are networks within tissue, there could be networks across tissues. And in this new concept of homogenic disease model, almost all genes could contribute to a complex disease, but there are important genes like these hub genes in red that could be more important, and there are also peripheral genes that potentially are less important. So in our research, we try to incorporate these different dimensions into our projects so that we could truly understand the complexity behind each of these diseases and across different diseases. All right, so, so the overall research directions, I partition them into this genetic aspect, the environmental aspect, and the therapeutic aspect. So from the genetics perspective, we wanted to understand among all the hundreds or even thousands of genetic risk factors uncovered for individual diseases, what are the network structure behind this? And can we find disease-specific and shared networks affected by genetic risks? On the environmental side, we have been studying diets, obesogens, injury, exercise, etc. And also of the idea of understanding the genetically affected networks and environmental affected networks could help us guide the identification of drugs or even nutritional factors that could reverse the disease networks. So in the middle of this would be methods, because to integrate all kinds of data is no easy task. So along the way, when we need it, we also develop some approaches. So I first start with the method side. I'll quickly introduce two tools we have been developing. So one is Mergeomics, the other one is Pharmacological. So the Mergeomics aims to look for the disease networks, and the Pharmacological aims to look for agents that could reverse disease networks. So this Mergeomics pipeline has been published. Basically, the idea is when people study different diseases, looking at the different layers of molecular data, you're conducting association studies between each layer with the disease. For example, if you look at DNA variants, you are looking at genome-wide association studies or Jivas. If you look at epigenome, you are looking at epigenome-wide association studies, and you know, all the other layers are similar. So what you're getting are the molecular signals that are associated with the disease. What we could do is take all these different signals and overlay them onto gene networks that are constructed in a tissue-specific fashion. And for example, one part of the network could be informed by the Jiva's signal, and the other part could be informed by the transcriptome signal, and others could be affected by the informed by the protein level signal. So the idea is if we could merge all these different layers, molecular signals, we could find some converging signals, pathways and processes that are influencing the disease. So this is available as a R package and also as a online web server. So if you click on this link, go to the web server, you could upload your data and look for converging networks associated with the disease. More importantly, it will help you identify the key driver or key regulators at the center. So you may ask, what kind of networks could we use? We use a variety of different network models, from co-expression to Bayesian to protein metabolic networks. So all this could be incorporated in the analysis. And this was developed across years by a postdoc and two former PhD students. And the second tool I wanted to talk about is this Pharmacological tool. This is the effort from two graduate students, Yin-Wei Chen, a current graduate student, a former graduate student, Doug Arneson. So there are other drug repositioning tools out there, but they mainly use cancer cell line as the model, and then treat the cancer cell lines with various drugs. And we thought that that could be a little bit less physiologically relevant, so we decided to look for tissue and species-specific drug signatures. We initially focused on FDA-approved drugs, but we're expanding it to other drugs that are under testing. So basically, we collect all publicly available data that can inform on the gene expression changes by various drugs. And we collect different tissue data and different species, focusing on human, mouse, and rat. Then we could build a database where we know in each tissue, in each species, which genes are going up, which genes are going down. And coupling with the disease network that we get from the Mergeomics side. So if we know there are networks connecting these red ones are the disease genes, and if we know the drugs target the same similar network, that could be a good match for treatment. So it's again available as a web server. It's not formally published yet, but we do have it available. So these tools have been, we have been using these tools in various applications to understand different diseases over the years. So these are some of the most recent applications. So I will focus on some applications in on the genetic side. So how we use those tools to identify genetically perturbed networks of individual diseases and shared between diseases. So although this is not really directly relevant to the brain, it's just a form, serves as example. So for example, for this non-alcoholic fatty liver disease, we took the Jivas and the tissue-specific gene regulation information and model the networks. We could find the tissue-specific pathways and the networks, and we could prioritize novel regulators that weren't discovered before for fatty liver disease. And we could also, this is in collaboration with Dr. Jake Lucy's lab. His lab, postdoc Karthik, knocked down our predicted key drivers to show that they indeed affect fatty liver disease. And also using our Pharmacological tool, we could predict which drugs could target non-alcoholic fatty liver disease. And these are the two of the top drugs that we predicted, and a postdoc in the lab has been validating the drugs. At least one of them shows a very strong effect. Another one is kind of intermediate, but it also shows a trend towards reducing non-alcoholic fatty liver disease. And going to the brain-related diseases, we have been trying to look for the connection between Alzheimer's disease and type 2 diabetes. And AD, as you may know, we're nowadays calling it kind of type 3 diabetes. So it's tightly connected to metabolic disorders. And so what we did was collected a lot of data from the genetic studies of Alzheimer's disease and also type 2 diabetes studies from various cohorts, and also used the Mergeomics to connect them to tissue-specific networks. So we could identify this core network that are shared between diabetes and AD involving metabolic pathways and immune functions. Again, using our Pharmacological tool, we could predict which drugs are targeting this particular network. So we have a long list of drugs that we could test to see whether that could reverse AD or not. So I'll also briefly touch base on the environmental side of our study. So here we have been looking at various environmental factors. High fructose, this is mostly in collaboration with Dr. Fernando Gomez-Pinilla here at UCLA. And we have been also looking at other diets and obesogens and injury, etc. So for all these environmental studies, we also take a multi-tissue approach. So both tissue and single cell level, different brain tissues involved in the disease could be affected by the environment, and also peripheral tissues looking for cross-tissue interactions. So also not only multi-tissue, we also take the multi-omics approach, looking at the transcriptome, epigenome, metabolome, gut microbiome, etc. I don't have time to introduce in detail these studies, but I wanted to kind of use this slide as a guide to move into this from tissue to single cell studies of complex diseases. So over the years, we have been applying this new single cell transcriptomic technology to understand various cardiometabolic diseases and also various brain disorders from traumatic brain injury, Alzheimer's disease, or ALS. Over the past few years, we have been publishing a few papers in this area. So I will focus on just going through one of the studies on traumatic brain injury. So we have worked with Fernando's lab on this mouse model for TBI, and we conducted single cell study across 3 different tissues, the hippocampus, hypothalamus, and also the peripheral blood cells. And so overall, profiling, tens of thousands of cells, and we could identify tissue-specific cell types and cellular responses to brain injury. And also we look at 2 different time points as well, from acute to chronic time points, so that we can understand the dynamic shifts in the genes and pathways in traumatic brain injury. So as an example, this is hippocampus single cell data at 24-hour, and the blue dots are the cells from the sham animal, and the red dots are from the TBI. You could see that for certain cell type, for example, the TG granule cells, you see a big shift in the cells from this TBI, from the sham animals. And in comparison to the cells from sham, and because their transcriptome are shifting a lot. And based on this analysis, most cell types seem to be quite sensitive to traumatic brain injury. This was led by a former PhD student, Doug Arneson. So we could also look at each individual cell type and ask which pathways are affected and what kind of genes are affected. So we could understand cell type specific molecular processes. For example, you see this metabolic depression pathway showing up in astrocytes and also in the CA1 region. A lot of metabolic related genes involved in these cell types. And obviously you find other cell types that are have genes important for amyloid development, etc. And we could also model cell-cell interaction networks using this kind of single cell data. For example, we could compare the cell-cell interaction networks between the control and the TBI to find there are some, shifts in the network organization, for example, between the astrocyte and the neurons. This is the initial connection in the controls, but after the injury, there is enhanced communication between the astrocyte and the neurons. So we can also use this type of data to guide the identification of targets. For example, based on our single cell study across many cell types, across different tissues, we find that this TTR is a brain thyroid hormone transporter, is upregulated at 2 different time points, only in the different cell types in the hippocampus. So, but not in the frontal cortex or blood. It has very strong tissue specificity. And this is a transporter for thyroid hormone. Thyroid hormone is important for regulation for metabolism. So we decided to treat the injured animal with the T4. This is the brain form of thyroid hormone to see if we could reverse the TBI phenotype on memory and learning. It did, and we could show that the T4 treatment could reverse the genes that were changed by TBI back to a comparable level to the sham animals. We could also show that the treatment mainly worked on modulating a lot of metabolic processes. So from this kind of single cell studies, not only we could understand more mechanisms, we could also identify targets. There are additional single cell studies, for example, this one with Dr. Scott Chandler and Sharmila Venugopal in our department. We have been studying this ALS between the SOD1 mutant and Y type, and we could identify cell type specific genes and pathways that are altered that could serve as potential targets. Similarly, we have been doing multiple studies on Alzheimer's disease. This is led by Jessica Dean, a graduate student in the lab. So on the mouse side, we have been using the 5-time FAD animals to study multiple tissues, multiple cell types, and look for the cell type specific mechanisms. On the human side, we have been working with Dr. Milan Fiala to look at the blood leukocytes, because these are the samples that's accessible from the patients. Interestingly, in both studies, we try to incorporate dietary modulation. So we treat the animal models and also human subjects with supplements to see whether they could reverse the cellular changes. I don't have time to go into detail with that, but that's the direction we are going. So just to, again, summarize that we have been using multi-tissue, multi-omics approaches, to understand the genetic and environmental risk factors and the how they impact the disease development across a variety of diseases. And we have been developing tools to also look for therapeutic interventions. Okay. So the take-home message here is that this type of multi-tissue, multi-omics integration can help identify complex tissue and cell-specific disease mechanisms. And these studies could help us have a high-resolution systems level maps at the molecular, cellular, tissue levels, and to help us find the key regulators. And also this can help us guide the selection of new therapeutic agents to normalize disease networks. With that, I wanted to thank the various contributors to the projects I mentioned today, from the Mergeomics, Pharmacology, the fatty liver work, AD to type 2 diabetes work, and the TBI and AD and also ALS work. Without the graduate students, postdocs, and undergrads, and also our fantastic collaborators, none of this would have succeed, have been successful. So thank to all of them. And here are my lab members. I particularly wanted to thank them for their hard work over the years. Thank you very much, and I'm ready for questions.
Thank you, Sha. That was very fascinating. So I just put the chat up for everybody. If you want to ask questions, either type them into chat window or just unmute yourself or raise your hand. But unmuting probably would be the easiest, and just asking. But I'll follow the chat. If there are questions, please ask them. So maybe I can lead off with one. Mm-hmm. So you addressed it a little bit in the last slide. So how comparable are these networks that you're finding between different organisms? So there's always, mouse is a bad model, mouse is a good model. So do you think that there is sort of, that there's consistencies or not at all?
Oh, yes. So there are definitely a lot of consistencies. We have been doing this mouse to human comparative studies. What we find is about 70% to 80% of sharing at the pathway level. But if you look at the individual genes, they may not directly overlap as well. And sometimes the direction of the change could be different as well. But overall, if you look at the pathway or network level overlap, the consistency is fairly good.
So in those that you looked at, the hubs would be similar. So the hubs that you're targeting pharmacologically, that you get those savings in animal models, would say, those are good targets in humans too, at least good ones to try.
Yes, I think at the hub level and also their neighborhood, mainly in their neighborhood pathway and network structure level, the consistency is very good. But if you look at just the individual genes identified from mouse and identified from human, there would be some differences.
Right.
Yeah.
So there's one question to ask, how many species have you compared? So sort of expanding on what I just said between mouse and human. So have you maybe outside even of a human application disease, if you just look from I don't know, very early on, up to monkeys and invertebrates and so on, do you find consistencies there?
We haven't done that level of comprehensive comparison yet. So far, the main comparison has been at the mouse and human level. But the one main direction for the lab in the future is to expand the species coverage to really look for even compared with worms, fruit flies, so then to mammals. But it depends on the data availability and the disease specific studies. We do, our focus is look for disease mechanism. Sometimes, certain organisms probably, you don't have a direct disease measurement, or the data type may not be kind of perfectly matching, or lack of data on certain species. Yeah.
So as you said, fruit flies would be a very interesting one, right? Because fruit flies and worms are used as model organisms. So
yes.
Are there any more questions? Are you free for collaborations?
We have been a bit of saturated, but we're trying to accommodate more and more.
Yes, I'm sure that a lot of people, you'll get a lot of emails after this to say, hey, could you look at my network? So anyway, so thank you very much. That was very fascinating.
Thank you.
We'll move to Ziva Cooper. So Ziva, just please take it over.
Great, thank you so much. And let me just go ahead and share my screen. Okay, so today I'll be talking about the work that I've been doing over the last several years related to understanding the effects of cannabis and cannabinoids by using a controlled human behavioral laboratory. And people here might have heard me talk about pain before and how cannabis and cannabinoids might be helpful for pain reduction. But today I thought I would take a different slant and talk more about abuse liability and how cannabis and cannabinoids can impact the abuse liability of other substances that are abused. So many of you have heard that a lot of people believe that cannabis can be used as a medicine for up to 700 different conditions. You can see 68 indications listed here. Of course, the newest indication for the use of medical cannabis is for prevention of coronavirus. So we have yet to have good data about that. But there's been quite a bit of buzz related to the potential for cannabis to be able to be used for substance use disorders. About 3 years ago, there was compelling findings from observational studies suggesting that cannabis might be helping to wean people off of crack cocaine. And also with the opioid pandemic, we are seeing news releases all the time about how cannabis might be helpful in either substituting for opioids for pain relief, or actually helping people that have opioid use disorders wean off of their opioids. So today I'll be talking about some of the data from our lab that directly address this issue. And before I do that, I just want to talk about cannabis and what it is. So cannabis is a plant that has over 140 unique constituents. The unique constituent that we know most about is tetrahydrocannabinol, THC. We know that the intoxicating effects of cannabis are due to THC. We know that THC from the cannabis plant can have negative cognitive effects. We know that THC has abuse liability. We also know that there are certain therapeutic indications for which THC is helpful for, including pain, increased appetite, and decreased nausea. THC in most plants and in most products sold now is usually the most abundant cannabinoid. And we know that THC is a partial CB1 receptor agonist. And when we look at cannabis's effects in the laboratory, we usually attribute those effects to THC, since all the other cannabinoids are taken out of the cannabis that we look at. Other cannabinoids that we hear a lot about include cannabidiol, CBD. It's all over the place. Now plants are being bred to be CBD dominant and have less THC. And there are other cannabinoids that are of interest as well that we'll start hearing more about. And there's some interesting preclinical literature suggesting the therapeutic effects of these other phytocannabinoids. So when asking the question if cannabis can actually be helpful in treating substance use disorders. It's interesting to look at the preclinical literature where there's a fair amount done looking at CB1 receptor agonists such as THC and full CB1 receptor agonists and its effects on the reinforcing effects of opioids. And there's some literature showing that CB1 receptor agonists can actually decrease the reinforcing effects of opioids in non-human primates, specifically self-administration of opioids. Across the board, if you look at all the evidence in non-human primates, it seems like acute administration of CB1 receptor agonists does not increase the reinforcing effects of opioids. It either does not affect the reinforcing effects of opioids, or it actually decreases the reinforcing effects of opioids. If you look at the evidence related to the impact of CB1 receptor agonists on cocaine self-administration, so models of cocaine abuse liability in animals, it's much more mixed. In 1999, there was a compelling study with THC in rodents showing that acute administration of THC was able to decrease cocaine self-administration. But since then, the literature has been quite mixed, where in adolescent animals, it looks like THC and CB1 receptor agonists might actually increase cocaine's abuse liability self-administration, whereas in adults, it has a different effect. These effects differ across CB1 receptor agonists based on agonist efficacy. So there's a lot of conflicting evidence there. In addition to conflicting evidence in the preclinical world, when you look at observational studies and epidemiological studies, there's also a lot of mixed messages. When we look at state level data, it looks like in states that have medical cannabis laws, where there's increased availability and accessibility of medical cannabis, there seems to be a general decrease in opioid use in those states, as well as fatalities. However, in other studies that track people over time, there doesn't seem to be a reliable association between cannabis use and decreases in opioid use. In fact, some studies actually show that people who are using cannabis and use opioids over time show increases in opioid use. Same thing for cocaine use. When we look at observational studies in the population, there was that 2017 study that showed that people that use cocaine and use cannabis as well show a reduction in cocaine use over time. But more recently, there was a study that showed that cannabis use actually predicts relapse to cocaine use in people that are in treatment for their cannabis use disorder. So how do we figure out how the preclinical findings fit into these epidemiological and observational findings? And one thing to do is use the controlled human laboratory, where we bring people in and under double-blind placebo-controlled studies, we look at the effects of cannabis with THC on the abuse liability of these different drugs. And here we're talking today about cocaine and opioids. So for these studies, we have a very controlled environment where participants come into the lab and they're there for about 8 hours. They don't have any outside stimulation, so they don't have their phone, so they're not chatting with people, which might augment their mood. Their other activities during the laboratory session are controlled, so we can tightly look at mood. These people are cannabis users as well as cocaine users in the study where we're looking at combinations of cannabis and cocaine, or in the study where we look at cannabis's effects on opioids abuse liability. There are people that have had experience with opioids. And we look at the abuse-related effects of the drug combination. So again, how does cannabis affect the abuse liability of cocaine, or how does cannabis affect the abuse liability of opioids? And I should note here that you might be able to imagine the regulatory hurdles that we have to go through to do these studies. And it's true, these studies take a very long time to get off the ground because of restrictions from the FDA, the DEA, and also even getting a source of drug is an important hurdle that we have to overcome. So I'll talk about two studies briefly. The first study is just looking at the abuse liability of smoked cannabis with THC only in it, so minimal to no CBD or other cannabinoids, and its effects on different doses of smoked cocaine. And in the second study, we're looking at the effects of this cannabis, the same cannabis that we're using in study 1, on prescription opioid abuse liability. So for the first study, we looked at cannabis with 0% THC, so that's our placebo cannabis, so it has no THC, no other cannabinoids. And we also looked at cannabis with 5.6% THC. And this was smoked cannabis, and participants essentially got the equivalent of about 35 milligrams of THC during each session. After participants smoked cannabis, we had different cocaine conditions where participants smoked 0 milligrams of cocaine, so a placebo dose of cocaine, 12 milligrams or 25 milligrams, either when cannabis, the placebo cannabis was given or the active cannabis was given. And to look at abuse liability, we had a very simple measure where we just asked participants to rate on a visual analog scale that was 0 millimeters from no effect being 0 to maximum possible effect being 100 millimeters. And we asked participants to rate how high they were and if they liked the high and if they liked the drug effect to get at this abuse liability. We also look at pharmacokinetics to understand if the interaction between the drugs that we're seeing in behavior are driven by a pharmacokinetic effect. But I won't be touching on that today. So for this study, when we asked participants to rate how high they felt after they smoked different amounts of cocaine, we can see here that there was a dose-dependent increase in intoxication after smoking cocaine. 12 and 25 milligrams produced significant increases in high compared to placebo cocaine. When we combined this with cannabis, we saw a slight increase for the 12 milligram dose of cocaine when cannabis was smoked, as we see in the black bars. Next, we wanted to know, well, they're getting high, participants are getting high. Is this a good drug effect? Do they like this drug effect? So we asked participants, is this a good drug effect and do you like this drug effect? And we did see a dose-dependent increase for both ratings. And when we combined cannabis with it, we got some weird interactions here with dose, where cannabis, active cannabis with THC, increased the abuse liability ratings for 12 milligrams, but we saw a slight decrease with 25 milligrams, but that was not statistically significant. Next, we looked at cocaine craving. Now it's interesting, cocaine craving actually goes up when people smoke cocaine, unlike with opioids. Usually when people use opioids, opioid craving goes down. In the case of cocaine, what we see is that when participants smoked increasing doses of cocaine, 12 and 25 milligrams, we see increases in cocaine craving. When we look at cannabis's effects on cocaine craving, we actually see a modest reduction in craving associated with 12 and 25 milligrams of cocaine. So we found, as expected, that cocaine produces drug liking, increases in subjective value, and increases in cocaine craving. We did not see cannabis to significantly alter cocaine intoxication or these positive drug effects, such as drug liking, wanting to take the drug again. And we also saw a modest reduction in cocaine craving, which was interesting in light of the study that was published in 2017, showing that people who use cocaine show reductions in crack cocaine use over 3 years. The next obvious question to ask is, well, it looks like cannabis decreases cocaine craving, but what about cocaine self-administration? What about self-administration, or what about relapse? The next study we looked at was the impact of cannabis on opioid abuse liability. And for this study, again, we used cannabis with no THC or cannabis with 5.6% THC, delivering about 35 milligrams of THC. And we used 2 different strengths of oral oxycodone, 2.5 milligrams oxycodone, as well as 5 milligrams of oxycodone. And again, we asked people to just rate their intoxication and the abuse liability of cannabis and the opioids. So under placebo conditions, where participants received 0 milligrams of oxycodone and no THC, we saw no increases in intoxication under that condition. But as expected, cannabis with THC increased readings of intoxication. And oxycodone did not further increase that intoxication, 2.5 milligrams or 5 milligrams of oxycodone. For abuse liability, we asked participants to rate how good of an effect they were getting from the cannabis and how willing they would be to take the cannabis again. And as expected, under the cannabis condition where they were smoking it with THC, we saw increased ratings of good effect and take again. And the 2 doses of oxycodone did not alter cannabis's abuse liability in this paradigm. Next, we asked participants to rate how good of an effect they got from the oxycodone, from the capsule. I'm sorry about my dog. And what we found is that under placebo conditions, where participants were not getting active cannabis, 2.5 and 5 milligrams of oxycodone did not really produce measurable increases in good effect. However, when participants smoked active cannabis before receiving oxycodone.
Hey, Ziva.
Yes.
Is he applauding the results or criticizing them?
I apologize. Sorry about that noise. So what we found is that, and I'll just try and speak over my dog, is that we found that cannabis increased the abuse liability of low-dose oxycodone. Same thing for liking. We found an increase in oxycodone liking with active cannabis. So to summarize, oxycodone did not increase cannabis abuse liability or intoxication, but that's not really the question that we're asking here with respect to, can cannabis help with the opioid epidemic? We did find in this respect that cannabis did not decrease oxycodone's subjective ratings of abuse liability, although we didn't really see profound ratings of abuse liability in this study of oxycodone. We did see that cannabis actually increased oxycodone's abuse liability. So the next question again is to see how cannabis augments oxycodone self-administration. And we have a grant to look at this right now at Columbia University. So the take-home message, and my dog clearly wants to chime in here, is that as we're evaluating what we're seeing in the media and what we're reading in the preclinical literature and the human literature, I think that controlled administration studies, such as what we're doing here, are really important in understanding how cannabis can impact abuse liability. And these controlled studies of the direct effects of cannabis and cannabinoids, so here I just talked about one cannabinoid, THC, and we're looking at CBD right now as well. Controlled studies are really important to bridge the gap between what we see in the preclinical literature as well as what we're hearing about from epidemiological and observational reports. So with that, thank you very much. To be happy to take questions.
I just had to find my unmute button again. So thank you, Ziva. That was fascinating, and I actually really appreciated the comments from your dog. That was great. It's just nice on Zoom, I think, to hear a little bit about each other's
life, right?
That's right, absolutely. So that was great. There is one question that I see here in the chat from Catherine Cahill. Are there any human PET studies to show whether the combination of cannabis with cocaine or oxycodone increases dopamine release in the striatum?
I don't know of any off the top of my head that look at acute drug administration and its effects on acute drug administration of the combination of these drugs and its effects on dopamine as far as imaging studies go. And this is a big gap, I think, in the research right now, is that because it's so difficult to do studies, to do acute administration studies with these types of controlled substances, there isn't very much in the way of combining neuroimaging with acute drug administration. And I think that, here, especially at UCLA, there's great potential because we have expertise in the neuroimaging world and we have the ability to administer drugs and be able to combine both of these techniques for a really, powerful and impactful outcome.
Hey, Ziva, it's Chris. So is there any evidence for cannabinoids changing the metabolism of oxycodone? I mean, it's you know, all these zip enzymes, et cetera, really, and with all these different compounds in cannabinoids, you've got many chances to inhibit the metabolism or change the metabolism.
Yeah, and so this is a really good point, and this is why I think it's important to couple the pharmacokinetic piece with the behavioral outcomes. So what I didn't show you here is that for the cocaine studies, we did see that cannabis actually decreased cocaine plasma levels, which might contribute to differences in cocaine craving that we saw. We didn't do pharmacokinetic analysis for the opioid study. I know that other labs have looked at combinations of cannabinoids and opioids, doing pharmacokinetic analyses in laboratory animals, not so much in humans, but it does differ based on the opioid that you're studying. So that's a really important consideration to take into account.
Yeah, I guess the other thing is it depends if it's chronic or acute, right? Because
yeah,
if it's chronic, you're going to upregulate some of these enzymes and downregulate others, and you know, it's a big old mess. So
right, and Chris, you raise a good point about the population that's being studied. You can imagine, and we know that chronic cannabis use has an impact neurobiologically, where we see reduced CB1 receptors, or, you know, based on neuroimaging studies, it's been suggested that we see reduced CB1 receptors and differences in opioid receptors. And so how does that influence the behavioral outcomes that we're seeing in these laboratory studies? So when we choose the population for these laboratory studies, we have to be very careful and figure out, are we going to recruit people that are heavy cannabis users and heavy opioid users, and make sure that we are consistent across the board for the participants that are enrolling in that study.
So are you doing genetic testing for metabolism enzymes and seeing if there's differences? Because I mean, yeah, I mean, everyone's going to have you know, these are very polymorphic parts of the genome, right? And I'm sure everybody has a very different spectrum of metabolism. So I wonder how you can control for that in all these studies.
Right, and we haven't started collecting samples for genetic analysis. I think it's also an important aspect to consider. But, you know, if you do look at pharmacokinetics of cannabinoids in general, you do see lawful changes across participants. But for these interactions, again, I think that, you know, looking at genotype, but also looking at the pharmacokinetics, so you can actually say, is this happening in the person? You know, can we attribute the behavioral changes that we're seeing to differences in pharmacokinetics will be important.
Yeah, good. Okay.
That's sort of a follow-up question to that, maybe follow-up, I don't know. So you're giving your study is on these, you know, purified, very well characterized prescription level drugs. How, you know, how does this compare to street level drugs? And how, as Chris said, you know, maybe you're adapted to street level drugs, but now you get something totally different into your system that in a way your system has never seen before. Is that a concern?
Well, so this is a particular issue when we're talking about cannabis. And, you know, there is this idea that because cannabis has hundreds of different chemicals that people hypothesize interact with each other. You know, the stuff that we're studying in the lab, where it's just cannabis with only THC, doesn't generalize to what people are using in the natural ecology on the street. And I think we're trying to get at that as well, to understand different cannabinoids. So I just presented data on THC. Now we have investigations where we're interested in looking at other cannabinoids, either in conjunction with THC or individually. So we can tease apart some interesting pharmacology there with respect to the whole plant.
So there's one more question on the chat, which says, thanks. Is prolactin involved in cannabis interaction with dopamine? How about calcineurin? There's this background here that I do not understand.
Right, and it's been a lot, sorry about my dog. It's been a long time since I thought about that, and I'll have to look look back. I appreciate the reminder of the context for that question. So
Okay.
I'm not sure off the top of my head. Thank you.
Okay, excellent. If there are no more questions, I would like to thank both speakers today, Shah and Ziva, and I'm sure that if you have a question that just comes up, they'd be happy to answer it even offline. So thank you for the contribution of your dog. I think it really was awesome, and thank you both for speaking. For everybody, please remember that we're going to continue to have joint seminars at noon for a couple of weeks. So the Magoon lecture is next time, and then we'll hear from all the BRI term chair holders, or almost all of them, in the subsequent weeks. So please stay tuned. Tune back in next Tuesday on the same channel. Okay, thank you. Thank you all.
Thank you.